I do think we are heading towards a cure.
We're getting there. I always tell my patients in my lifetime, we're going to do this. We absolutely are going to do this. We've seen too much change in the last decade or 2 decades to not reach that point.
Hi everyone, my name is Cindy Chmielewski and I've been living with multiple myeloma since 2008. Prior to my myeloma diagnosis, I was a fifth grade teacher, and since being diagnosed with myeloma, I realized how important it is for patients to be part of their care and educated so they can be part of that decision-making process. So now I'm using my teaching skills to help myeloma patients learn a little bit more about myeloma so they can engage in discussions with their doctors. And now I would like to introduce Dr. Landgren is a hematologist oncologist with the University of Miami's Sylvester Comprehensive Care Center. He is a professor of medicine, the chief of the myeloma division, and a co-leader of the translational and clinical oncology program. I remember when I was diagnosed with myeloma back in 2008, there were very, very few treatment options. Most of the things available today were not available. Induction therapy, that first therapy was normally a doublet. Eventually it became a triplet. Now we're going to talk about triplets or quadruplets. Maybe we first could talk about, like, what do you think about if they're transplant eligible or ineligible, newly diagnosed versus relapsed refractory? Like, what are those considerations?
Well, I think that a big kind of step forward in the field has been the evolution of all the different options. So it's no longer, in my opinion, a matter of picking A or B. I think we have pretty much a whole alphabet to present. So there are very many options, and it starts in the newly diagnosed setting. There are a lot of options. And if someone has a recurrence, again, there are very many options. Looking at the guidelines that dictate what we're allowed to prescribe, there are over 40 different combinations that are in this guideline in the setting of relapse refractory disease. So when I have a newly diagnosed patient, if I'm thinking, is this person a potential candidate for transplant or not? I think that has also shifted over the years, because we can technically transplant many more patients than we could before. We can transplant people that are older, and even if there are comorbidities, it's possible. On the flip side, I personally think that the need for transplant has probably gone down because there are so many new treatments.
So, moving on, now that we know a little bit about clinical trials, let's talk about some specific clinical trials for newly diagnosed myeloma patients. Newly diagnosed patient is the patient who's just starting treatment, and in my mind, I always thought that clinical trials would be for later on down the lane, maybe when you're running out of treatment options. So this idea of clinical trials in the newly diagnosed was new to me, but it makes sense. And Dr. Costello, can you talk about some, like, maybe newly diagnosed trials that might be of interest to the audience?
Yes, of course. And I think you're so right, Cindy. It's so true that clinical trials exist at every part of the journey with myeloma. And it really offers opportunities for patients. Yes, you're either getting what the standard of care is, or you're getting something that might be better. And so it gives patients opportunities to the next new and cutting edge thing. I think the last few years we've had the benefit of some of the outcomes of some of these newly diagnosed trials, and particularly the ones that finally are getting published. So the Griffin study looked at, as you mentioned previously, Cindy, two drugs are good, 3 drugs look better, maybe 4 is the best. And so it looked at to say, hey, when someone's newly diagnosed, our usual kind of cocktail is 3 different drugs together, but what if we give them 4? Does that have better outcomes, more likelihood of achieving remission? Does it get us more durable remissions where people are staying in remission longer? And so we finally have some data now that's finally the final analysis that was published that showed us that those patients, when they're first diagnosed, who get 4 drugs, have deeper and more durable remissions than those who just get our standard of care, which our standard of care now seemed, is outdated. Our standard of care are 3 drugs we were doing until about 3 years ago. And so now it's really just in the recent couple of years, maybe, that we've said 4 drugs is the way to go. And this is specific to our, let's say, younger and or healthier patients, because age is just a number, but the reality is that some intensive therapies may be too much for a patient, depending on their health or other medical issues that they may have. Now, so I think if we found what that next new and exciting thing is for someone who is maybe younger and or healthier, the next question is, well, what about someone who may not be as young or as fit or possibly more frail? And we have finally said that the triplet combination that came out of the Maya study combined 3 different drugs, which we proved again was better than 2 different drugs, to say that though that cocktail truly has exceedingly effective outcomes for patients who may be older or more frail, for example. So I think there's a lot of work that's been done to get us to this point. I think we'll talk about it in a sec about what's coming next, but we're just so glad to see that we finally have a new standard of care that we can have subsequent and future trials use as our baseline to compare to for the next steps to get us to that cure.
Yeah, we're in exciting times, and we've been talking about how things have changed. I know when I was diagnosed with myeloma back in 2008, the standard of care induction therapy was just a doublet. You used 2 drugs. Now the standard of care is a triplet, but it seems to be moving towards quadruplets. And before we start talking about quads and what we think about the benefits of some of these 4 drug combinations, can we just focus a little bit on some of the clinical trials that are triplet combinations and which ones have been gaining your attention and why?
I think that in the more modern era, the bigger triplets that I pay attention to are the RVD regimen or VRD, depending on which letter you put first. So this is bortezomib, lenalidomide, and dexamethasone. So that is one of the 3 drug combinations that I think are important. They have pushed the field forward. I was involved in the development of the KRD regimen with cofilzomib and lenalidomide and dexamethasone. I worked on this back in 2009, 2010, 2011, so more than 10 years ago. And the difference between these 2 regimens is the proteasome inhibitors that different. So you have less of the neuropathy. You have no added neuropathy with cofilzomib, which you have with bortezomib. But if you give cofilzomib to a patient with underlying cardiovascular disease, you could push that patient into congestive heart failure. But I think that's a great, great combination. And for patients that are otherwise fit, that would be my go-to therapy. Then you have the DRD, which is daratumumab or Darzalex with lenalidomide dexamethasone. That was approved in 2019. That has been a game changer. It takes away all the problem with bortezomib neuropathy again, and you replace the proteasome inhibitor with an antibody. So giving a CD38 targeted antibody with lenalidomide dexamethasone, that has really changed the field. And I would say that it has also erased some of this age discrimination that's been in the field for a long time, where patients that were younger could get the more effective therapies and they could get the chemotherapy with transplant, with melphalan, while patients who were a little bit older and more frail, they used to have very few options. I think the addition of immunotherapy has really, really made a huge step forward.
Just how about your thoughts on the trials that are doing like selinexor dex against elopalm dex? Any thoughts on that trial?
Yeah, so those are combinations and trials that are available for patients who have relapsed or refractory disease. Elotuzumab is also developed in that same space. Elotuzumab is a drug that binds to a protein called CS1 or SLAMF7, and it was discovered many years ago. And in the laboratory, initially, it was found that this antibody bound to myeloma cells and they died. But in patients, the elotuzumab drug alone has not really been found to have any clinical benefit. But this has been partnered with either with lenalidomide or with pomalidomide, and if given in combination, that has been found to be more efficacious than just giving lenalidomide alone with dexamethasone or pomalidomide alone with dexamethasone without the elotuzumab. So elotuzumab adds to these backbones, if you want. And then you mentioned the selinexor. You can use that in different combinations. It has been approved with bortezomib, and it's approved for patients with the first, second, or third relapse. And there are other combinations as well. You mentioned with pomalidomide, and there are also with carfilzomib. Selinexor is a new class of drugs. It binds to a protein that is sent into the center of the tumor cells, the nucleus. So it's a blocker of certain proteins that go in and out of the nucleus. And that's been found to work very well also in myeloma. So that's another good option. I think we need more classes of drugs because if you treat the patient and it works, that's fantastic. But if the therapy stops working, we need to have other classes of drugs. So these are great options you bring up.
And let's just talk about one more class of drugs before we move on to the four drug combinations. Blenrep or belantamab mafodotin. We had it on the market for a bit, and then it was pulled because it didn't meet one of its primary endpoints. But it's still in trials, like the Dream 8 trials are using Blenrep in combination. Can you talk a little bit more about Blenrep?
So Blenrep or belantamab mafodotin is a antibody drug conjugate. So what that means is that it is a small molecule. It's a toxin similar to, for example, lenalidomide or carfilzomib. That is a molecule that can have anti-myeloma effect. But the difference between belantamab mafodotin and these other drugs is that the way it's delivered. So the drug is not just given as a tablet or an injection or an infusion. It's bound to an antibody, and that antibody is then infused into the vein. And when the antibody comes into the body, in the human body, it looks for the corresponding protein that this antibody is being designed to bind to. And that is the BCMA protein that's widely expressed on the surface of myeloma cells. So these antibodies, they find the myeloma cells, they bind to this BCMA protein, and once they have done it, they sort of knock on the door. The myeloma cell says, "Who's there?" and lets in this antibody. And the antibody comes inside, and when it's in there, it releases this toxin, and the toxin will kill the myeloma cells. So it's like a Trojan horse. So this is the first and so far only antibody drug conjugate developed for myeloma. As you pointed out, Cindy, it was approved in 2020. So, and then it was pulled from the market in 2022. But they have kept it on the market, and in agreement with the FDA, the drug is available for compassionate use. And the fact is that the drug works for many patients. So, if there is a doctor that thinks this is the best drug, the best option for a given patient here and now, that doctor can reach out to the FDA and get permission to use it. And lastly, there are other trials that were ongoing that they had already launched with other combinations with belantamab mafodotin, and there are two of them ongoing. So there is a control arm, and now it's belantamab mafodotin in combination. So the study I mentioned that failed was a single drug belantamab mafodotin versus a control arm. But there are two trials that use combination therapies versus control arms, and they are ongoing. Yeah, so they are part of the so-called DREAM program. The belantamab mafodotin was initially developed in DREAM 1, and then there's been 2 and 3 and 4 and so forth. So now we're up to DREAM 7 and DREAM 8. And the trials I mentioned that are still ongoing use belantamab mafodotin. One of them uses it with bortezomib dexamethasone, and the control arm is bortezomib dexamethasone. And the other one uses it with pomalidomide and dexamethasone, and the control arm is pomalidomide and dexamethasone. So unlike the trial that failed for the confirmation after the initial accelerated approval, that used just single drug belantamab mafodotin, but now it's in combination.
Now let's talk about four drug combinations. Do you think that the four drug combinations are going to be the new standard of care for the newly diagnosed myeloma? And if so, which four drugs do you envision to be used as part of that induction therapy? And how do you weigh those risks and benefits of adding that other drug with the side effects it may cause?
Sure. So I think that Four drugs are definitely here to stay, but not for everyone. I think we talked about before the importance of personalizing this approach and how we take into consideration so many different aspects of you, your health, your myeloma when we make these decisions. So, well, all things being equal, I do think that four drugs are here to stay. The Griffin study showed us that it is better than three. Now, there's more trials, though, that are coming out. We talked about the different phases of trial. That Griffin trial was a phase 2 trial, which showed us important things. But we really like to extrapolate that data to say, let's use it in several hundred different patients to prove what we think truly was shown in the Griffin study. And not only to say that it works, which was the point of the Griffin, to say, let's compare patients after some finite period of time to say, how likely are you to achieve a remission, for example. The next studies that are coming along, like the Perseus phase 3 trial, for example, is going to show us how we can get that durability of the response and prove that the initial study Griffin was really right and really is the proof that the quadruplet or four drugs is here to stay.
So that's a very good question. So where are we? Where are we heading? So I think similar to most things in life, there will be people that are early adopters, and then there are people that are like the very last people to change their mind. And some people, they build a career on just saying nay, 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 or no, no, no all the time. So that's how the field sort of goes. Yeah, so the three drug combinations I mentioned before, proteasome inhibitors with IMiDs and steroids, and I mentioned that the evolution has also been CD38 targeted antibodies with IMiDs and steroids. So when we talk about four drug combinations, it's basically a merge of those. So that means that you add a CD38 targeted monoclonal antibody, and the studies that are out there are using, for the most part, daratumumab, and that's added to the proteasome inhibitor, IMiD, and the steroid. So in plain language, what you're talking about are DARA RVD or DARA KRD. Those are the two out there that have been presented. And there are three trials that have so far been published using those combinations. But as you heard me saying, it's not only the four drug combination up front that's different between the two arms. It's not 4 versus 3. It's also the maintenance being very, very different. So you could, of course, ask the question, what happened if you gave everybody 3 drugs to begin with and just had a 2 or a 1 drug maintenance? Would that make a difference? And that study has not been done, so we don't know the answer to that. The other combination is the DARA KRD. And the first trial to be published, I was the lead investigator. It's called the Manhattan study, and I did that when I was at Sloan Kettering. And we gave the DARA KOD for 8 cycles. Patients were not transplanted on that study. And after 8 cycles of treatment, patients have the option of collecting their stem cells or not. And actually, they did it after 4 cycles. And after 8 cycles, they had the option of being transplanted or not. And some patients were MRD negative and they chose to go for transplant. But the majority of patients that were MRD negative, they chose not to be transplanted. So they kept their stem cells in the freezer, and we have kept on following these patients. So far, we have not really seen any significant difference whether you were transplanted or not. But the study was not designed to fully answer this question. The third and the last study that has used the DARA KOD regimen is the MASTER trial. And that was developed by Luciano Costa. And he treated also a smaller number of patients. There were 81 patients treated. Patient got 4 cycles of DARA KOD. They were transplanted. And if they were MRD negative after each of these 2 steps, they were taken off therapy. If they just turned MRD negative, they got another 4 cycles of DARA KOD. And again, if they were 2 times in a row negative, taken off therapy. And they could get additional 4 cycles. So they could end up with a total of 12 cycles. And every patient was transplanted in this study. He took patients off study of treatment if they were MRD negative 2 times in a row. And he has shown that with a relatively short follow-up, that patients that didn't have adverse cytogenetics continued to be free from disease after 1 or 2 or so years, while patients that have more aggressive disease, their disease would flare back again if you take them off therapy. So that's kind of where the field is right now.
Dr. Costello, let's talk about some of these newer immunotherapies. These were things that were, I don't think when I was diagnosed, they might have been a twinkle in a researcher's eye, but I don't even know if they were in clinical trials. CAR T cell therapy. I remember when I first heard about it, being from New Jersey and Philadelphia is right up the road. I heard when the University of Pennsylvania and CHOPS did their first CAR T cell patient and how well that the response was. So, can you talk a little bit about maybe first for people who are not familiar with what CAR T cell therapy is, what exactly that is? And what are some of the benefits of using maybe this type of immunotherapy in treating your myeloma? And what could some of the challenges be?
This has been, I think, one of the single most exciting treatments I have seen kind of in my career. And I remember the same thing, Cindy. I was in New York City where they were developing CAR Ts for other kinds of cancer patients. And watching a patient go through it was none of us really knew what to expect. But I'll tell you, over the course of the last decade or so, we've learned so much. Let's talk about it. So, CAR T stands for chimeric antigen receptor T cell therapy. That's a lot. So, your T cells, I think it's important to recognize, are part of your immune system. Your T cells are responsible to be kind of these fighter cells that when your immune system identifies a bad guy, they say, hey, T cells, I need you. These are the bad guys, come fight. So they are designed to look for, you name it, number of bad guys that kind of come into your presence, whatever that may be. What if there was an opportunity to train your T cells to specifically look for your myeloma? So speaking of personalized medicine, right? It doesn't get much better than this. This is your own immune system fighting your own cancer. So every myeloma cell has little tags on it, if you will, that identify it as a myeloma cell. In the most recent times, we have found that there's a tag called BCMA, B cell maturation antigen. This is one of the tags on the myeloma cell that says, hey, this is me, I'm the bad guy. The CAR T cell is a way that we can train your T cells to look for that tag. The logistics of it can be a little overwhelming sometimes, and it is one of the challenges of CAR T. But very basically, the logistics include, if you've ever undergone a stem cell transplant or have heard of it, it feels very similar. Your immune system T cells are removed from your body, similar to like a blood donation, if you will. They're sent off to a specific company then who re-engineers them, basically does genetic modifications in order to create and train the T cell to look for that tag we talked about. Once those cells are engineered and made, they are mailed back to your doctor, and your doctor, with some additional logistics, will basically put them right back into your body. Your immune system now trained to look for your myeloma cell. And so that is a very, very basic overview with lots of kind of logistics and details and risks associated with it, but has really offered a very unique means of using your immune system to fight your own cancer. So the benefits that we've seen, there are two different CAR Ts that are approved by the FDA in 2023 for the use of treating relapsed and refractory multiple myeloma. It's approved for patients who have had 4 or more different treatments, meaning that your myeloma has come and gone, let's say at least 3 or 4 different times, has received at least 4 different kinds of treatments to make it go away. And now you're looking for your next treatment option. The benefits of it is that it is a one and done treatment. Now, for all you myeloma patients, and Cindy, I know you can talk about this, you have probably been on myeloma treatments for a very long time. And those, they're great, right, Cindy? It keeps a lid on everything. Maybe it can kind of delay it from coming back, but with that comes cost, comes time, comes side effects, comes a lot of additional challenges. And so one of the things about CAR T is when you receive this treatment, that is the treatment one time. And your immune system now is trained to go in there, kind of go to war against your myeloma and knock everything down again. So it's a very exciting opportunity to allow patients to have a treatment-free interval where you've done your treatment and can enjoy maybe years without any additional therapy. Now, unfortunately, it's not curing totally everybody yet, but at least it is buying us time, right? Which is what we really do with myeloma treatments, is kick the can down the road until the next CAR T comes or till the next treatment comes. So lots of exciting benefits of it. There are, with new drugs come new side effects that your doctor and you need to learn about and figure out how to manage to keep you safe. With new exciting options, there's a lot of popularity around them. And with this, which is not your doctor going to write a prescription and just hand it to you next week, there's a lot of logistics involved in getting the T cells out of you, engineering them, sending them back to you, having the manufacturing capabilities to do that. And that is time, that is money, and that is accessibility. And so there is still not enough access for our patients, for everybody to have it who needs it. And so I think there's lots that's going to come. We're going to continue to fine tune and hone this. But from what we've seen so far, it's been a next new wonderful treatment option for our myeloma patients.
Can you talk a little bit about what we found out from the Carditude trials and maybe specifically Carditude 4 and what's happening in Carditude 5 and 6?
So the Carditude trials, though, are just what you said. They are looking at how can we use Carditude with Somatitude, right? Like they are looking at the cilta-cel product, which is one of the two CAR T products targeting BCMA, that tag on the myeloma cell. The FDA approved it kind of right in the middle of COVID, right? We had two CAR Ts approved and we were all sitting around just tweeting at it and excited from our own couches, but it was approved for patients who had had 4 or more prior lines. So we all kind of sunk in our chairs saying, but I have to wait for it. And as we've talked about kind of the clinical trial phases, the way that drugs get approved is they get approved on the basis of safety and effectiveness from the very get-go. And truly, maybe unfortunately, but truly for some patients, this is oftentimes studied with patients, as you say, who have no other options because we need to prove that it works. Once we prove it works and that it is safe, then we can kind of open up our net, cast a wider net, if you will, to see, well, heck, if it works that great at that point in the disease, why wait? Could we potentially study it in patients who have been diagnosed with myeloma more recently or have had fewer than 4 treatments, maybe 1 or 2, or maybe even when they first diagnosed? So the Carditude trials, and there are a lot of them, are looking at exactly this. How can we take what is our current standard of care, not a placebo, things that we now use when you're first diagnosed or first relapse or second relapse and you're looking for your third, your next line treatment? Can we compare those things and say, this is what we usually use. We know it's effective. And if it works, let's make a randomized trial. Let's make a trial that says, I'm going to give you, patient, what I know works. I'm going to give you, patient, this CAR T cell, which we know works in this other group of patients, but let's see if it's as good or if not better than what our usual suspects are. And so a lot of these Carditude studies that are happening now is to say exactly that. Is it at least as good, if not better than what our standards of care are? And so now, once we've said in the relapse setting, I think the thing we're all next going to ask, and I'm sure is coming next, is we've been doing bone marrow transplant a very long time. And it has been great in a sense that it works. It has its issues, it has its side effects. There are parts of it that are no fun, but everyone's starting to say, can we compare that to CAR T instead? And so I think we're going to see more and more data coming out about some of these Carditude studies. There are KarMMa studies, which is evaluating the second or the other BCMA CAR T cell with ide-cel, it's called, asking this question to say, all right, we have a newly diagnosed patient. Half of you are going to go to transplant. Half are you going to go to CAR T? Let's see what happens. So we can finally start to answer the age-old question of, do we still need transplant? As a disclaimer, I'm a transplanter. I believe in transplant. I think until someone shows me it's better, there's still a great role for transplant, but I can't wait to see how those results turn out.
Now let's talk a little bit about the bispecific antibodies. They too are a new type of immunotherapy. Can you first talk a little bit about what the bispecific antibodies are, maybe their mechanism of action, how they work, and what are some of the side effects that we're seeing from the bispecifics?
The bispecific antibodies are, from a development point of view, very different from the other antibodies. We talked about daratumumab and elotuzumab before. They were developed back in 2015. So the first bispecific antibody that came to myeloma was in end of October of 2022. And then we have additional 2 bispecifics in August of 2023. These bispecific antibodies, what they do is that they bind to the myeloma cells on the surface of the myeloma cells, and then they also bind to the surface of T cells. And what that means is that they hone in on the myeloma cell and they hone in on the T cell, and they bring these two cells to sit next to each other. So when the T cell sits next to the myeloma cell, it will kill the myeloma cell. So when we talk about CAR T cells, what does that mean? Well, then we take out T cells, we make them into CAR T cells. So what that means is that they are T cells that bind to myeloma cells. So you could say it's sort of very similar. So instead of taking out the T cells, which is what we do with CAR T cells, we keep the T cells in the body, but we give an antibody that links the existing T cells in the body to the myeloma cells in the body. So we are kind of trying to engage the T cells with the myeloma cells, so the T cells can act as the treatment for myeloma. Yeah, so you also asked me about the side effects. So the side effects for the bispecific antibodies are quite similar to the CAR T cells in that regard, that you have the so-called cytokine release syndrome, and you could also have these immune cell mediated neurological syndromes with neurological side effects that are due to these T cells. So they are similar in that regard, but usually with the bispecific antibodies, you have less profound, less severe reactions. Most of the cytokine release syndromes are what we call grade 1, and some are grade 2. But we very seldom or never almost see higher grades. But with CAR T cells, you could see grade 3 or grade 4. And grade 1, 2, 3, and 4 is basically a scale to grade the severity of it.
And with the bispecifics, I know they're having some, I guess, higher rates of infection. Can you talk a little bit about that?
So we see these type of reactions, but we see less profound of them. And then you ask me about the infections. So infections can happen both in CAR T cells and with the bispecific antibodies. And what I've seen when I reviewed all the papers, and I continuously go through the literature and I talk to other investigators, is that I see that the infections are more likely to happen earlier on in the CAR T cells because you have more profound suppression early on. But with the antibodies, because you keep on giving them the way they are developed, they're given for the most part once a week, and then they can be turned into every other week. And there are new trials looking into give them, say, once every month or so. But the fact that you keep on giving these drugs, that also means that you not only hit the myeloma if it's there or keep the myeloma away, unfortunately, you also keep on hitting the immune system. So you will see there is some continued immunosuppression, and patients seem to be more able to capture infections over time with the bispecific antibodies. Now, there are ways to to prevent this, and this all comes back to the physicians. I always emphasize that the doctors have to know exactly how to manage this. So a way to avoid this, to prevent these things, is to give intravenous immunoglobulin G or IVIG.
Sounds good. Now, the bispecifics that are currently approved are approved as a monotherapy or just by themselves. There are some trials like the Redirect trial and the Monumental trial that's doing some combinations. Can you talk a little bit about those trials?
Yeah, the field is moving very fast forward, and there is a whole range of combinations. So, to touch on some of them, what was found some years back ago now by Damian Green out at the Fred Hutch Cancer Center in Seattle, he found that the BCMA, that's the target for many of these bispecific antibodies and the currently available CAR T cells, that the cancer cells, the myeloma cells, can protect themselves by chopping off the BCMA from their surface. So the drug binds to the target, but then the target gets chopped off. So it's circulating free in the blood, and the cancer cells are just swimming by, waving to these receptors and the drug. So that's not a good scenario. So how could you prevent that from happening? So what Damian Green and his colleagues found was that this happens when the cells release something called gamma secretase. So they used gamma secretase inhibition inhibition to stop this from happening. There are newer developments now, and we have been part of this as well. And I work closely with Damien. He's a long-term friend and colleague of mine. And I think that it's probably overkill to give every patient this. I think there are some patients where it still is maybe the right thing to do, but as a general platform, I think probably not needed. So that's one line of investigation. We are not yet done, but that seems to be the way the field is going. Then you could increase the efficacy of it. You could use, say, other antibodies. So you could use daratumumab in combination with bispecific. So this is almost like if you're at home and you are getting hungry and you're thinking I should really start preparing dinner, and then you open your refrigerator and you see, what do I have? I have a tomato here, and I have a cucumber, and I have maybe some fish. Or so then you start thinking how you can combine all these things. So that's how these trials have been developed. People have daratumumab, they have teclistamab. Let's put them together and see what happens. So there's quite little scientific rationale why these drugs were picked. They just picked them because they had them. And also people have used teclistamab with, say, lenalidomide, or elranatamab, which is also an approved BCMA bispecific antibody, together with lenalidomide and also with pomalidomide. And then you can start using it also with proteasome inhibitors, with carfilzomib or other drugs. Or why not taking two of these bispecifics at the same time? So you could take a BCMA and a GPRC5D targeted bispecific antibody and use that together. There is data coming out, and this ASH will present a lot of this data. We will see. I think the bottom line at this point is that these single drugs are amazing. The bispecifics in patients that are heavily pretreated, 4 or more prior lines, on average, it's about 60% of patients responding. And to put it in context, when daratumumab was approved in 2015, it was 30%. So they are double efficacy. And it's not only double because in 2023 versus 2015, patients today have been treated with many more drugs when they go on trials compared to almost 10 years ago. There were virtually no drugs around. So you could argue and say patients today that reach that point on average are sicker than they were 10 years ago. And still it delivers over 60%. So that's amazing. But you can also see with these combinations that you can bump up these response rates from 60 to 70 to 80 to 90 to 95%. Also, these drugs would probably become part of the frontline therapy, because what we have known for a long time is that what works in later line usually works earlier, better in earlier line. So I think if we have this discussion in 5 years, Cindy, probably everything has changed.
Yeah, exciting times, very, very hopeful. And I hope to be here 5 years from now. You know, when I was newly diagnosed, 5 years from now was really scary to me. For now, with all these new drugs, I'm counting on being and having a discussion with you in 5 years. So let's talk about this word cure. I know a few years ago, maybe about 7 or 8 years ago, when I was attending some of these scientific meetings, doctors were very hesitant to use the word cure. They were talking about controlling myeloma and keeping it, at bay for several years, but I never heard the word cure coming out of their mouths. But now, more recently, I am hearing the word cure, and that doctors are thinking that maybe we are curing some patients. Dr. Costello, can you talk a little bit about cure in myeloma and maybe some trials and your perspective on that cure in myeloma?
Of course, Cindy. I think this is the other C word, right? The big C we think about is cancer, but realistically, the big C, that cure word, is becoming something we're really starting to flirt with, as you say, over the last couple of years here, as we've seen, just like you said, just kind of this explosion of new medications that are out there. As you mentioned, in the last 2 decades or so, we have seen the amount of options for treatment go from nothing, essentially, a transplant, for example, to just this wide variety of options that are letting people get into remission and stay in remission for very durable, long periods of time. And now we're starting to see that happening for such long periods of time. We're saying, well, is it ever coming back? So I think we still have work to do to understand the difference between a long, durable remission, which means it goes away and stays away for some great period of time, versus a cure, which means it ain't ever coming back, which is what we're all aspiring to. And so I think we're getting there. I always tell my patients, in my lifetime, we're going to do this. We absolutely are going to do this. We've seen too much change in the last decade or 2 decades to not reach that point. But to answer your first question about, what are the right treatments, I would argue that this is not a one-size-fits-all type of cancer. Although multiple myeloma is kind of grouped together as one kind of form of a blood cancer, there are very different subtypes and very different kind of forms of myeloma that will act differently. And some are kind of, let's say, easier to treat and make go away than others that can be a little bit more stubborn. So when we're thinking about what the ideal treatments are for any individual person, I think it has to be exactly that. We have to look at you as a person, you as in your health, you in your social situation, your support, for example. And then we have to look at the biology of the disease and kind of put all of that together to figure out what are the best means of achieving that cure that you're talking about.
I do think we are heading towards a cure. And I also want to point out and say the reason that the field has not talked about that topic in myeloma in the past, I think, is for multiple reasons. One is that, quite frankly, the drugs were not good enough. We used to have 1 or 2 drugs for many years, way before I started practicing. And then there was the emergence of high-dose chemotherapy with melphalan, the so-called transplant. Then there were the rediscovery of the IMiDs with Bordology's discovery from thalidomide, and then that led to lenalidomide and pomalidomide. We had the proteasome inhibitors coming just around those drugs were coming out, and then we have had all the immunotherapy. So the field has evolved, and I think we can see very many patients achieving remissions, and very many patients these days can also achieve MRD negativity. So there is no detectable disease. And I want to also lastly say that I have many patients in my clinic that are cured. And I have patients who have been off treatment for actually 20 years, and they remain off treatment doing very, very well.
Yeah, we're in exciting times. And just like you said, if you ask many myeloma specialists, they would have a different answer of what a cure is. I think the same holds true for patients. If you ask what they expect in a cure, you might come up with different answers. But to me, it sounds wonderful being treated for a set period of time and then being off drugs and, for an extended period of time. That would be my definition of a cure.